Hello, everyone. Welcome to part two of this three-part lecture series on microbial growth. This chapter covers the different factors that affect their growth, needed chemicals, physical requirements, uh, what do we need for them to grow on in the lab, and so on. So let's get started. This is part two. We were looking at chemical requirements, and the last one I finished up with part one was on trace elements. Let's look at oxygen now. So I want you to look at this picture here that shows the different growth of bacteria, and these are test tubes that have agar in them, solid agar. And what you can do is you can insert the bacteria into this agar, this nutrient agar in these tubes. They're called deeps, is what they're called. And you can remove bacteria and put them on the end of a wire called a stab, and you can deposit them into the agar. And then here are five categories of bacteria, and they've been categorized according to their utilization of oxygen. So over on the left, there are obligate aerobes. So of course, in reality, they don't look like this. It looks like, I don't know, a bunch of grapes or something floating in this yellow stuff. What you would see with an obligate aerobe is if you took the bacteria and you stabbed them down into the agar, let them incubate for 24 hours, and then look at the tube the next day, you'd see the bacteria growing along the top of the agar. The area, the line where you stab the wire down into the agar, you could barely see it or even see it at all. And the bacteria would be spilling out through the top, the hole in which the stab made in the agar. They would spill out of that hole and then grow along the top of the agar. This is kind of showing it like they're growing down in the agar a little bit. That's not the way it looks with an obligate aerobe. They want to get out of that agar as quickly as possible. They need to get to the oxygen. That's why they're called obligate aerobes. They are obligated to have air, or in this case, oxygen. So they need the oxygen in order to survive. They have to have it. Without it, they die. So they want to get the heck out of that agar as quickly as possible so that they can then get up to the oxygen, and then they'll spread across the surface of that agar in the test tube. Next category, going to the right, facultative anaerobes. So these are bacteria that they can survive in both environments, with or without oxygen. Now they do better with oxygen. So if in the case of this one, if this were what you'd be using a facultative anaerobe in real life, you take the bacteria, stab it into the agar, you'd actually be able to see the stab line that you put into the agar when you put the wire down into the agar, because the bacteria, they they want to get out to the oxygen, but they're not all that concerned about it because they're surviving just fine down deep in the agar. They do better on the top, and so you will definitely notice growth of facultative anaerobes on the top of the agar because their metabolic rate is it's enhanced. I mean, it increases some whenever the oxygen is present, but they don't have to have it. So they like oxygen, but it's not necessary. Obligate anaerobes, that would be the opposite of an obligate aerobe. Obligate anaerobes, they cannot tolerate oxygen, so when you stab them down into the agar, they're going to grow down at the bottom as far away from the oxygen as possible. So whenever you take the wire back out of the agar will kind of like seal up around it, and then they will just grow on the bottom, and the growth appears cloudy. So the agar it takes on kind of a cloudy appearance down at the bottom. Nothing growing up top. If anything happens to venture up towards the oxygen, those cells will die. Those are the three most common. The next two are not very common categories at all. There are very few bacteria that are aerotolerant anaerobes, and there are very few that are microaerophils. So aerotolerant anaerobes, the name kind of tells you what it is. They can tolerate air or oxygen, but they're more anaerobic. So they're kind of like the opposite of the facultative anaerobes. Facultative anaerobes, they do better in oxygen, but they don't have to have it. Aerotolerant anaerobes can tolerate air, but they're going to do better in an anaerobic environment. Microaerophils, so this one, if you split it into three parts, they love air, but in small amounts. Micro, small, aero meaning oxygen, and fill they love. So they love oxygen, but only in small amounts. So they're going to grow kind of in the middle, like this picture is showing here. They don't want too much oxygen, but they've got to have some. So they're going to stay away from where there's an abundance of oxygen, but they can't go too far down in the agar, or they won't get any oxygen at all. So they're going to grow somewhere in the middle. So here's the definitions of them, what I just said. So you can look at those. I just mentioned all these, so let's keep moving on. Organic growth factors. So we're talking about these compounds that are necessary for them to survive, but they can't synthesize them. They can't make these compounds. So they get it from the outside, from the environment. Vitamins, amino acids, nucleotides. Most bacteria, they're able to synthesize all of their own vitamins, but they don't have the necessary enzymes to do so, some of them. So they have to get these compounds from somewhere outside. So they have to get it from a host. And so this makes them parasitic in that way, because they're going to take something from their host in order for them to survive. And so we call them organic growth factors that are necessary for them, but they're unable to produce them. So let's look at culture media. Media is plural, medium is singular. This is what is necessary for the organisms to grow on. A culture medium, nutrients prepared for microbial growth. And so in the lab, there's a variety, but by far for a micro lab, the most common would be Petri plates and test tubes with agar in them. The term sterile, obviously no living microbes are present, so sterility becomes an important factor in a micro lab because you don't want cross-contamination and so forth. And so everything needs to be sterilized, so an autoclave is essential for a micro lab. An inoculum, introduction of microbes into a medium. So when you inoculate something, you introduce the microbes into the agar. And culture, as we said at the beginning of this chapter, microbes growing in or on culture medium. So they can actually grow within it. There's a way to prepare agar where you have the bacteria actually living down in the agar itself, like for example with those tubes we just talked about, but you can do that with Petri plates as well. It's a different technique, but there's a variety of ways to encourage their growth. That's what this is all about. So what are the requirements for media? Correct nutrients for the microbe, so this becomes a little bit specific. I mean, you can just use general nutrient agar and just about any bacterium will grow on it. Sufficient moisture and oxygen, if oxygen is needed at all. If you're dealing with an anaerobic bacterium, obviously oxygen's, you're not going to be using that or needing that. Proper pH. And as I said, the proper pH range for bacteria is about 6.5 to 7.5 for most species, but of course it depends on what you're culturing. You may need to alter that pH. And it has to be sterile. There can't be any competing organisms because then you won't be able to research the particular organism that you want to research. There's going to be other organisms that will have contaminated the culture. So agar, as I said, number one medium that's used for culturing bacteria. It's a complex polysaccharide. It comes from a marine alga. There's a species of alga that's growing in the ocean, and it grows in abundance. And so it's harvested, it is dried up, it's chopped up, it's pulverized, it's put into a powder form, and then it becomes a solidifying agent. The bacteria don't eat the agar. Generally not metabolized by them. That sounds so much more educational and scientific, doesn't it? Generally not eaten by the microbes, so they don't break it down. So when you put bacteria on a Petri plate, the agar becomes the surface on which the bacteria will live. And in the agar are the nutrients and the water content and whatever the bacteria need to grow. But they don't consume it. If they did, then it would be useless because you'd try to culture bacteria in the agar, but the bacteria would eat it, and then you'd end up with this soup or whatever would be left with it, depending on how they could metabolize the agar. So they don't have the enzymes to break it down. It works great. And there have been synthetic agars that have been used in the past and are still used today. They don't seem to work quite as well as the natural agar that comes from the marine alga. It has numerous uses. In the culinary world, it's used as a food thickener, and say, for example, in ice creams and other foods, so it thickens products, and it works well there. It's also used as a laxative as well. So it has more uses than just for culturing bacteria, but for this course, and probably its number one reason, is it's being used for culturing bacteria. Now, there are two basic types of media. There is chemically defined media and then complex media. Chemically defined media, they use the exact chemical composition is known, mainly used for experimental work. So you know exactly what is contained within chemically defined media. With complex media, what you would find in that are extracts and digests of yeasts, meat, or plants. Now, what exactly is an extract or digest of yeast? I mean, you could figure out what it is exactly, but you don't need to know with complex media. It's not necessary. Composition varies from batch to batch, and it's used in intro micro labs. We call it nutrient broth if it's in a liquid form, meaning it doesn't have the agar in it to solidify it, or nutrient agar, then, which is the solid form. So to help you understand that a little better, let's look at two recipes here. The one on the left is chemically defined, and the one on the right is complex. So if you look at the chemically defined one, you'll see that everything that is in this medium is known. The quantities are given, but on the left side of the recipe there, you can see exactly what's being put into this medium. On the one on the right, the complex medium, you have peptone, which is partially digested protein. Well, it doesn't say exactly what protein it is or what amino acids are contained within it. Beef extract, don't know exactly what that is. So these are ingredients that we don't know the exact composition, but it's not necessary to know the exact composition. All we know is that when we use complex medium, the bacteria like it. And they grow really well on it. But if you want to culture a very specific bacterium, or you want to alter the medium in a very specific way to see what the effects are on the microbe in question, the one you're experimenting upon, you would use a chemically defined medium for that. So complex medium, just general, growing, whatever. Chemically defined, you're getting into particulars, research, and so forth. Reducing media is what's needed to culture anaerobes. Contains chemicals that combine with oxygen to deplete it from the media, and then heat is usually used to drive off the oxygen to remove all oxygen and get rid of it entirely because anaerobes cannot tolerate any oxygen present. One very simple device to use is an anaerobic jar. It's got a lid on it that you can clamp down and tighten with a screw. It's got a rubber gasket seal around the top of it to prevent any oxygen from getting in. The oxygen in the jar and the hydrogen combine to form water on the surface of uh, palladium, which is what is inside there. Those catalyst pellets, they will combine those two, so and that will then lead to the removal of the oxygen from the internal environment. And you know that the oxygen has been depleted whenever the methylene blue indicator is colorless. So you put your Petri plates in there and then just clamp it tight, and then shortly the oxygen will be depleted and the bacteria then will be able to grow and thrive in this anaerobic jar. And they work well. You can't get a whole bunch of plates in at one time, but you can get a number of them. It's very realistic there, showing 6 plates. That's about typically how many you'd put in your average anaerobic jar. You can get up to, if you need a greater quantity and you get high tech and you got a little more money available, you use anaerobic chambers filled with oxygen-free gas, and you can see it has glove ports so that no oxygen gets in. Everything is tightly sealed up. The oxygen concentration is monitored very closely using sensors. And so then you can grow your anaerobic bacteria, and you can grow them in more quantity. And obviously you can experiment with this now because you can actually manipulate the plates, and you can do things with the bacteria here under the plexiglass container. So much more useful for culturing the anaerobic bacteria, and you can do it on a larger scale and you can actually do a little more experimentation with it as well, uh, being that they have glove ports and you can handle these. Capnophils are microbes that grow better at high CO2 concentrations, like in the intestinal and respiratory tracts and in the mouth, which can lead to periodontitis, or gum disease, so degradation of dental tissue down below the gum line. Um, these bacteria, there's a group of bacteria called fusobacteria that live down underneath what's called the gingival crevice. That's that kind of like that seal that is between the gum line and the tooth itself. Those bacteria will live down in there, and the CO2 concentration is higher there in those regions, the intestinal and respiratory tracts and in the mouth. So higher than what? Higher than the atmosphere. So the atmospheric CO2 concentration is around 1%. In those regions of the body, the CO2 concentration gets higher. It's not a whole lot higher, maybe 3 to 5%, somewhere in that ballpark. And so these microbes will grow better there because of the higher amount of carbon that's available due to the higher concentration of carbon dioxide. Can you get an environment like that so that you can experiment with these organisms? Well, you can get the very high-tech candle jar. You get a glass jar and a lid and a candle and put your microbes in there, light the candle, close the lid, and it increases the CO2 concentration to about 3%. Combustion reduces the oxygen concentration, and then CO2 concentration goes up about 3 times compared to what it is in the environment, in the atmosphere. CO2 packets, so this is one where you have a packet that has a seal on it. You open it up, put your plate in, and then you seal it. It's got a sticky seal across the top. There's a gas generator inside there, this tube, that will reduce the oxygen concentration and produces carbon dioxide. So it too will do you'll achieve the same thing as the high-tech candle jar, but I think this one might work a little better, but it's all going to turn out the same anyway. So you might as well go with your cheaper low-tech option, I suppose. So anyway, that ends part 2 of this 3-part lecture series on microbial growth. Please stay tuned for part 3, and thank you for watching.